Hello, and welcome to Brewing Brilliance, a podcast where we talk to academics from across the university who are involved with the Healthy Ageing and Chronic Conditions Research Institute. We will be speaking to researchers who are leading innovative, interdisciplinary approaches to promote lifelong health, delivering improved interventions to manage chronic conditions more efficiently. These sessions are essentially opportunities for us within the Healthy Ageing and Chronic Conditions Research Institute to meet colleagues from across and beyond our faculty to foster knowledge sharing, collaboration and networking. My name is Adrienne Cleverly, and today I am joined by Dr. James Murray, who is a biochemist with a career-long interest in ageing-related diseases. This includes diabetes, cancer and neurodegenerative diseases, with his research focusing on how cells repair damage. This led to a broader interest in the biology of ageing, and now he investigates how to enhance human health span. Hello and welcome.
Oh, thanks very much.
Thank you for joining us. So I guess I should really start by asking, how did you end up in this field of research?
Oh, good God, that is a good question because I didn't start out my career as a biochemist. I was an immunologist of all things. So interested in how the immune system is co-opted by viruses in the first instance. So my training is virology and molecular immunology. But then in the early 2000s, I went to the States and started working on protein biochemistry and a concept called reversible phosphorylation, which is essentially a mechanism by which cells can signal. So in sensing the outside environment and translating that signal to the inside of the cell to stimulate certain processes to happen. If it's an immune cell like a macrophage, stimulates it to take up foreign material or microbes and so on. So that's where I really got started into how diseases occurred. So that was work I had funded on diabetes research. And then I moved back to the UK in the mid-2000s, continued working on cell signaling, and in the context of diabetes, but then also having one eye on other types of diseases because the signaling pathways, the reversible phosphorylation control mechanisms seemed common to multiple diseases, and that included cancer as well as neurodegenerative diseases, but that came much later. So in the mid-2000s, I started working on cancer cell signaling and still doing some diabetes research as well. And the nexus for those diseases was that cells have to repair or eliminate damage. And by doing so, they can enhance their longevity, the health span of the cell. So everything I've ever worked on up until very recently has been cell-focused. So the pathways that I then started to research were called proteostatic pathways, ability to remove damaged organelles like mitochondria or damaged pieces of DNA or misfolded proteins. And so those pathways, and one of them in particular called autophagy, transcends most human diseases. So autophagy is involved in diabetes, cancer, Alzheimer's disease, Parkinson's disease. And so over a number of years, I started spreading thinner and thinner in the laboratory and with different projects working in all these different areas. And then I had one of those moments of epiphany where I realized that the pathway that I was interested in is also a pathway that decreases in its ability to do its job as we get older. So therefore, the biology of aging affects the autophagy pathway or proteostatic pathways. So that's when I realized that what I needed to understand more was how does that change as people get older or as an organism gets older. So that's where I am today, working in Swansea, looking at the mechanisms that control the biology of aging.
Wow. And so in a nutshell, that's how you got to where you are now.
In a way, yes.
So with leading that into healthy ageing, I guess that expresses why it's so important for research, because we are in a demographic older aging profile, aren't we? So
yeah, we have a lot of customers.
Yes.
So in terms of how my research translates into healthy aging, there's lots of information out in the world today because of the internet and social media proposing lots of things that you can do to live longer or to live healthier. I mean, the classic example is diet and exercise. That's probably the best thing that anybody can do to improve their chances of surviving for as long as possible, and is as healthy as possible too. Because most of our aging program is really dictated by genetics and environment. Okay. So what my lab is trying to understand is how the genetics or the mechanisms that go wrong or change as we get older influence our health span. Okay. So if I was to give one example, we know that autophagy, the pathway that I'm biochemically interested in, its activity tends to decrease over lifespan, particularly in the brain. And that's coincident with the development of lots of neurodegenerative diseases. So anything where proteins start to clump together, Alzheimer's disease, Parkinson's disease, autophagy seems to be implicated in that. So we want to try and understand why is it that autophagy becomes less proficient as we get older, as our system ages, the biology of our system. And while we don't know the answer quite yet, we think it's got something to do with how cells control the repair mechanisms. And this is an evolutionary concept that I'm going to touch on just very, very briefly.
Yeah.
So one of the many theories of why aging occurs, there's this theory of antagonistic pleiotropy. And in antagonistic pleiotropy, the sense is that the genes that give us an advantage to reach reproductive age, to reproduce, raise children, have families as humans, are also the genes that are kind of counterproductive as we get older too. So one of those key genes, key proteins, is a protein called target of rapamycin. And it's a key one to mention because there are lots of drugs that are now being developed, or natural compounds which are known to affect its biology, which seem to be improving lifespan, longevity in at least animal models, and some circumstantial evidence in human studies too. So we think by modulating the signaling pathways which control pathways such as autophagy, we can improve health span. But we're doing it sort of backwards because we have effects that we know happen because we modulate these pathways. So exercise will affect TOR signaling. Diet affects TOR signaling. Certain natural products will affect TOR signaling, and we see these aging longevity effects, but we just want to tease out the biological mechanisms to figure out exactly why that happens. And if that is happening, can we develop new interventions that can be used as accessories for treating patients or particular individuals in a more lifelong way, so that we have a natural way of adjusting our health span rather than having going to the extremes of medical interventions or strict diet and exercise regimens.
So trying to sort of move away a little bit from just the generic advice that they give you, which is just, like you say, focusing on healthy lifestyles.
Exactly, because although lots of people embrace healthy lifestyles, more often than not, it's a chore.
Yeah.
You know, don't have the chocolate bar, don't have a drink at the weekend. You need to be exercising 3-5 times a week. Lots of people find that difficult. Life gets in the way. And while I don't want a pill that fixes everything, we want to figure out the biology so that we can perhaps provide some ways of supplementing our health span.
So in that way, what makes your research unique?
No.
Because you touched on it a little bit, and I get the feeling there's more to that.
So my career has always been focused on cells.
Yeah.
So we've always wanted to understand what happens in those biochemical bags, which are what cells basically is. It's just chemistry, but in a living system. So what we've done is we've moved away from just using single cells or cell types to understand the biology, and we've moved into a model organism. Now, obviously, model organisms have their place in research. They're very important for various aspects of this process. But what I wanted to do was avoid a number of things. I wanted to avoid using higher organisms because I'm an animal lover.
Yeah.
So the idea of using mice in research is not something I'm keen on. We needed a system which was powerful enough to understand systems biology, so how different organs and organ systems interact with one another, but it's also something simple enough that we can tease out the biology relatively easily without having to use extreme expense or powerful technologies that require lots of research scientists to focus on it. And the other thing that we felt we needed was a system which, again, to understand lifespan, you do not want to be studying an organism that lives 30, 40, 50 years because they might outlive your career. So we had to pick an organism that had a long enough lifespan that we could see changes that are similar to humans, but short enough lifespan that you can do some work with it within a 1 or 2 or 3 year period of time, which is most funding cycles. And we also wanted to stay in a vertebrate system. So that's a backbone skeletal system. So what we chose was the African turquoise killifish, Nothobranchus furseri. And the reason why we chose Nothobranchus furseri for many reasons, but one of the key reasons is its lifespan is about 6 months.
Right, okay.
So it's a short-lived vertebrate fish. Okay, so similar to other animal models such as zebrafish, but zebrafish tend to live years, whereas these tend to live months. The other advantage to Nothobranchus furseri is that it actually does develop lots of the tissue-level pathology we see in older humans. It's the only vertebrate model where you can see evidence for neurodegeneration. We see sarcopenia, muscle wasting in the fish. They develop kidney problems. They may have heart problems too, which we suspect. They also tend to have fatty livers, so they tend to have fatty liver disease. So there's lots of the aspects of the killifish which are reminiscent of human pathology, and that's where a lot of labs across the world now are focusing their time and trying to characterize those age-related changes over lifespan in the killifish and map those to humans, so we can establish the killifish model as a bona fide model of aging that will help us unlock lots of new information.
And it can help you unlock it, like you've just stated, it gives you lots of different things you can actually look at, so it doesn't tie you down. But finding all these similarities to compare must have been a research on its own stand.
Yeah, but it's kind of funny how this all falls out, because the whole field of that focuses on using the killifish, the African turquoise killifish, only became established because of a renowned scientist called Alessandro Celerino, in Italy, who by chance had a friend who kept tropical fish, and his friend mentioned that he knew that Alessandro was interested in aging, but he mentioned to Alessandro, they have this tropical fish, but it won't live long, and it gets sick, and Alessandro immediately realized the impact of this, a short-lived species that develops pathologies that could be similar to human pathology. So that was an important first step. That was only in the early 2000s. So we're talking about a very new model. Maybe 20, 30 labs across the world are currently using it.
And we have one in Swansea.
And we have one in Swansea.
Which
in
is
the UK.
incredible.
And there's maybe 1 or 2 other labs in the UK that use this currently. But we're hoping that will expand.
And that sort of gives a great insight into how it can move forward as well, because it's already gone, like you said, it was the early noughties and it's already evolved and by a chance encounter of who someone's friend is.
Absolutely. But then most science is based on chance encounters or that's funny observations. Yeah.
The beauty of science, I
indeed.
guess. Yeah. Well, that in itself is very, very interesting. So thank you so much for taking the time to talk to us about that today. If you are interested and want to find out more, then please reach out to James. You can find him on Swansea Uni and you can look him up under his staff profile. Equally, if there are anything that you want to find, we do have the research available. So if you'd like to find out more about this podcast or the Healthy Aging and Chronic Conditions Research Institute, please visit the Swansea University website. Thank you very much for joining us. I've really enjoyed listening to your unique research. Please, like I said, if you are interested, reach out to us. I'm Adrienne Cleverly. This is James. Thank you so much.
Thanks very much, Adrienne.